Hi. Uh, I hope the PPT is visible and I'm audible.
Yeah.
Yeah. So in today's class, we'll just have a quick summary of all the lectures, brief contents, and we'll also go slightly into detail if there are any specific doubts in any specific topics. Yeah. So we start with basic overview of the course. So this course was intended to bring out the basic aspects of metabolic engineering, where stuffs including what is metabolic engineering, why is it required, what are the computational methodologies that were adopted, and how successful that was in identifying certain characteristics of a network, of a metabolic network, were discussed. And in the later stages of the course, other mathematical models, including 13-carbon metabolic flux analysis, were discussed, followed by CRISPR-based methods for introducing gene knock-ins or performing gene knockouts. And finally, case studies regarding uh, successful metabolic engineering approaches at the large scale were discussed. So in the first week, the main ideas that were discussed is the cycle which metabolic engineering follows, which is the design, build, test, learn cycle. So here in designing, based on previous knowledge, the organism is chosen, and based on the chemical of interest, the pathway is chosen. Then, in the build part of this cycle, the different parts, including promoters and genes, are identified and they are assembled either in the plasmid or in the chromosome. This particular part called the build is now called as synthetic biology. Further, once the design is conceived and the necessary uh, biological material, biomolecules are available such that the parts are built. Now we have to screen for the chemical of interest, and this is done in either of the two methods using high throughput methods or through spectroscopy. This particular part, called as the test part, is now termed as metabolomics, where the entire metabolome of an organism is analyzed. And finally, in the learned part, uh, the data collected so far is interpreted to improve the current strategy. And this is done by studying the flux through different reactions. This is where the metabolic flux analysis comes into play. So, as we all know, cell is a chemical factory, and by altering the genes, more chemicals can be produced. Metabolic engineering here is used as a directed evolution strategy, and particularly examples such as artemisinin from plants, where the genes for this was taken and it was inserted into yeast, and large-scale production of artemisinin could be achieved. Uh, with respect to the cell metabolism, it can be considered as a well-connected and complex network, and this network is identified from the genome sequence. So, in this first week, different examples were provided. For example, the way to produce succinic acid from the TCA cycle. So, one particular strategy that was proposed was increasing the enzyme responsible for conversion of pyruvate to acetyl CoA. But it doesn't help since only a fraction of acetyl CoA is going into succinic acid. So, this was a particular example that was discussed. So this gave an overview of what is expected out of metabolic engineering. After this, uh, the different uh, the way metabolic engineering has evolved over time was discussed. So there they say that, like in every other process, we want to improve the yield of our product of interest and reduce the yield of the undesired products. So metabolic engineering also evolved in in a way to increase the yield of our desired product of interest. In the first generation of metabolic engineering, microbes were grown and whatever compound it could produce naturally, that was extracted. In the second generation, this idea of mutate and screen was considered, where the mutagenesis was random. Because this mutagenesis was random, we were not entirely aware of the genetic and the metabolic profiles of the mutant strains. So it was not very well characterized. But in a way, through different screening methodologies, despite random mutagenesis, uh, strains that were capable of producing our desired compound with better yield were identified and they were scaled up. Camera off. Yeah. So, as I was saying, we were talking about the different generations of uh, metabolic engineering, and we were discussing specifically second generation where mutagenesis is random, which is specifically not desired. So, generation 3 overcame this random mutagenesis by introducing precise modifications of specific enzymatic reactions through genetic engineering approaches. Apart from this, in week one's lectures, the basic idea that metabolic pathways are integrated or interconnected was established. So, what was tried is uh, okay, an attempt was made to bring in the idea that because this entire metabolic pathway is interconnected or integrated, modulation and expression of genes should be coordinated for achieving our desired result. This idea was established. And, and, and the concept that by perturbing one specific variable in a metabolic pathway, there could be significant different effects. That was also established. An example of how the carbon metabolism in E. coli is strongly regulated by the presence or absence of oxygen was discussed, where in the absence of oxygen, uh, pyruvate doesn't get converted to acetyl CoA, whereas in the presence of oxygen, it gets converted to acetyl CoA and further goes down to TCA. So this was discussed. Apart from this, the basic elements of metabolic engineering, such as pathway design, pathway construction, and pathway optimization, it was introduced. Then, the idea that pathways should be assessed for converting a specific substrate to specific target product was also introduced. Here, it was uh, it was discussed that thermodynamic feasibility of a pathway and the flux within a particular pathway is important. And few terms like seed metabolites were introduced. With respect to enumeration, what it means is that suppose substrate A can be converted into product B by n different pathways. Let's assume that this is the case. Then enumeration means that we have to identify that which are all the possible pathways, the n different pathways which can give this product from the substrate. This is called enumeration. Whereas assessment is to further probe into the details of each of the specific pathway, which on the basis of a few parameters, which includes the thermodynamic feasibility, the toxicity of intermediate metabolites, and the expression of enzymes in hosts. Here, a particular web tool called BMICE was also introduced. Now moving on to week 2 lectures summary. So here we started with emphasizing the need for large number of genetic modifications. As we know that a cell contains the cell metabolism contains a large number of control mechanisms. If we try to alter a particular step by performing particular reaction, there could be ways possible that we haven't discovered yet that could undo what we did, which means that in order to engineer a cell to produce our compound of interest, it would mean that a higher number of genetic modifications are needed. Further moving on to the lectures, it was discussed that how sugars and other carbon sources are used for producing precursor metabolites. Now, precursor metabolites give rise to macromolecules and biomass, and they have this thing called bow tie structure, where large numbers of sugars and carbons get progressively reduced to produce 12 precursor metabolites from the central carbon metabolism. So, once this was discussed, it was also made clear that due to the tight regulation, redirecting the carbon fluxes in the central carbon metabolism towards molecules of interest is inherently difficult. And to further add on to this, it was also discussed how not in all organisms these reactions take place specifically in the cytoplasm. Uh, in certain in compartmentalized organisms, which basically higher eukaryotes or eukaryotes in general, TCA reaction, for example, happens in mitochondria, meaning that additional transport reactions will be needed if we have to rely on the flux of the precursor metabolites for producing our target molecules of interest. Once this was discussed about precursor metabolites, then the discussion was uh, the discussion was carried out on how metabolism can be divided into basically catabolism and anabolism. So catabolism is the reaction which involves basically the breakdown of large complex organic molecules into smaller and simpler molecules. Whereas anabolism is the part of metabolism where synthesis of complex organic molecules from simpler ones occurs. Then we try to link catabolism and anabolism, where how organic carbon gets converted into smaller molecules that gives rise to precursors, and precursors gives rise to macromolecules. Now, the part where organic compounds gives rise to smaller molecules is a part of catabolism, whereas smaller molecules getting converted into precursors and precursors to macromolecules, that is anabolism. And apart from these precursors, reducing equivalents such as NADH and energy currency like ATP is also getting transferred between catabolism and anabolism, making sure that metabolism is linked by catabolism and anabolism. Once this was discussed, then the overarching principles in living systems was discussed, which is first enzyme catalysis, where enzymes are needed for accelerating in both the forward and enzyme catalysis was discovered. So these can accelerate in both the forward and the reverse directions. And the way they function is by reducing the activation energy. The second principle is energy harvesting from redox reactions, where oxidation is used to harness energy in metabolically useful form. This is where the transmembrane proton gradient was discussed. The other principle is energetically coupled reactions, where coupling of a highly favorable reaction with an energetically unfavorable reaction would drive the energetically unfavorable reaction. Here, the idea of proton motive force and electron transfer was discussed. Then, further moving on, the framework of metabolism was discussed, where there are a set of fueling reactions which gives you the 12 precursors, and they give free energy for performing other reactions and the reducing power. Then we have the biosynthetic reactions, which are useful for for generating or synthesizing the building blocks, coenzymes, cofactors, and small molecules. Then the polymerization reaction, where macromolecules are being produced. Now, this macromolecule is being produced by uh, direct directed linkage of the activated small molecules. And finally, the assembly reactions, where chemical modification of macromolecules and transport of the macromolecules to specific locations within the cell, and also association of these macromolecules to form the cellular structure is achieved. So these four different kinds of reactions can be classified under the framework of metabolism. Further moving on, um, the idea that metabolism involves growth metabolism and maintenance metabolism was discussed. So in fueling fueling reactions, like we have different energy sources, different carbon sources, and different electron sources. In the energy source, we have chemo organotroph, chemolithotroph, and phototroph. Where in chemoorganotroph, the source of energy is organic compounds. Chemolitho, it's inorganic compounds, and photo, it's light. And with respect to classification carbon sources, it's auto and heterotroph, where if it's the source of carbon is CO2, it is autotroph, or if the source of carbon is organic, then it is heterotroph. And electron source, if the source of the electron is organic compounds, then it's organic, whereas if the source of electron is inorganic, then it is lithotrophs. Then the different metabolisms in uh, in a cell was discussed, starting with catabolism of sugar in the process of glycolysis producing pyruvate. Further, pyruvate gets involved in other processes like fermentation, TCA, anaplerotic reactions, which replenishes precursors, and also transamination pathways for amino acid biosynthesis. Now, for glycolysis itself, there are three different routes. One is the EMP pathway, where hexo-6 phosphate gets converted to pyruvate, and the pentose phosphate pathway, where pentose sugars, the 4 carbon sugars and the 7 carbon sugars are involved. And finally, the Entner-Doudner pathway, which is present in pseudomonas. This was also discussed. Yeah. In the next week, the idea of what we P by O ratio was discussed. He- So basically, we know ATP is the energy currency of a cell. And as ATP gets hydrolyzed to ADP and an inorganic phosphate, some energy is achieved. So we can consider that breaking of phosphate bonds is important for the uh, breaking of phosphate bonds is important for the metabolism of the cell. So P by O ratio describes the number of phosphate bonds for each oxygen atom used as an electron acceptor. Huh. Sorry, there was another internet issue. Yeah, so as I was saying, specifically 3 ATP phosphates are made for each proton that is transported by EPC for NADH. And for FADH, it is 2 ATP. Then the law of mass action was discussed, which states that the reaction rate is proportional to the probability of collision of the reactants. So let's say we have J moles of reactant A and K moles of reactant B, giving L moles of reactant C plus M moles of product B. Now, the coefficient K here will be given as C to the power L into D to the power M divided by B to the power K into A to the power J. This is the law of mass action. And apart from this, a way to represent equations to identify the rate at which mass is flowing was introduced in the form of ordinary differential equations. So let's consider a reaction S1 plus S2, which is a reversible reaction giving rise to 2 moles of product P. Now, d by dt of S1 and d by dt of S2, if we consider that this is negative V, which is the rate at which the mass is flowing, there is a negative sign here because both S1 and S2 are getting consumed and they're decreasing over time. If this rate is considered as negative V, then the rate at which product forms dP by dt is equal to 2V, because we already know from the reaction, the stoichiometries, that S1 plus S2 gives 2P. This was discussed. Further moving on, the different kinds of annotations that are generally used while working with metabolic networks was introduced. So 1D annotation is where sequence analysis is performed to locate ORFs and to assign function to the specific gene products. 2D annotation is 2D annotation means that despite or independent of assigning the function to the gene, we also have to identify interaction between genes. This will help us uh, get an idea about which genes perform what function and how coordinated is their function. This was discussed. After this, the concept of how signaling networks, which could be understood as stimuli or small molecules that bind to regulatory components, are the starting point for uh, a cell metabolism. So, signaling networks uh, give information to regulatory networks. These regulatory networks determine which regions of the pathway are active based on the signals that are received. Then, based on the regulation of the network, the entire metabolic pathway, which has to be either overexpressed or underexpressed, are then switched on or switched off respectively. Then, the breakdown of how from DNA sequence we get genetic circuits was introduced. So, so as we spoke about the annotations, from the DNA sequence, first ORF is identified, which gives rise to genes. Then ORF is assigned, which gives rise to the gene products. So from this is a part of the 1D annotation. After we have this, we try to do 2D annotation where we try to understand the coordinated function, which gives a structure to the entire network. And this entire network, if they perform one particular task, then they are called as a genetic circuit. So beyond this, regulatory networks were introduced. So there is a specific difference between a biochemical reaction network and a regulatory network. So metabolic networks in general are called biochemical reaction network, but statistically influenced networks are the regulatory networks, which means that they are based on transcriptomics, proteomics, and metabolomics. So here the models that are built involve protein-protein interaction, protein-DNA interaction, and DNA-DNA interaction. Whereas in a metabolic network, only the protein maximum that it is being involved is the protein-protein interaction, if at all. And the difference between metabolic networks and statistical influence networks were discussed. One major difference that we can remember just from the definitions is that because metabolic networks are at the base level involving specific reactions, they are most likely not to change once they are reconstructed from biochemical data. But statistical influence networks can change with additional data. As and when new information about a component that controls a particular reaction is available, it can uh, it can be introduced to understand or to modify the statistical influence network. Now, with respect to reconstruction, two different ways in which reconstruction could be achieved was discussed. One is the bottom-up data, which is derived from classical biochemistry, and it focuses on very few variables. An example that was specifically mentioned is the lacoperon and the galregulon, whereas um, in the top-up reconstruction, the components, links, and the network state for reconstruction are integrated and then validation is performed. And majorly, this is from high throughput data, not from classical biochemistry data. So then the reconstruction was divided into levels, four levels specifically. The first level where we know the input and the output. The second level, we have this uh, different sectors, catabolism, anabolism, and growth. In the third level, we further delve deep to identify which is the pathway. And the fourth level, the individual reactions. So once how the different components of reconstruction were discussed, then the stoichiometric matrix was introduced. So this is a sparse matrix, meaning that most of the elements are zero. This stoichiometric matrix is uh, usually defined as S. So it transforms the flux vector, basically the reaction rates, into a new vector that contains the time derivatives of the concentration. So it can be understood that S is a linear transformation of the flux vector. And apart from the stoichiometric matrix, two other matrix were also discussed. One of it is the elemental matrix, where we have uh, the elements of a particular compound in the in one axis of the matrix and the compounds themselves in the other axis of the matrix. And when the elemental matrix is multiplied to the stoichiometric matrix, since the reactions are mass balanced, this should equate to zero. Meaning, if 1 mole of an example of the same is if 1 mole of glucose and 1 mole of ATP interact, they should give 1 mole of glucose 6 phosphate and 1 mole of ADP. That's how mass is balanced. So this is one of the matrix. Elemental matrix is one of the matrix other than a stoichiometric matrix. The other matrix that could be constructed is based on the charge because for any chemical reaction, charge also has to be balanced. So beyond the stoichiometric matrix, two other maps were discussed. One is called the reaction map, and the other is called the compound map. So uh, these could be understood as converse relations. So in a reaction map, the way different metabolites are involved in a particular reaction is highlighted with the nodes as the metabolites. So any network has nodes and edges. Nodes are connected with each other through edges. In reaction map, nodes are uh, metabolites, whereas in compound maps, the nodes are the reactions themselves. This was also discussed as a part of week four's tutorial session, where we had tried to solve or come up with a reaction map and a compound map. So after this was discussed, a brief summary of network reconstruction was provided, and it is as follows. So we have to use information from databases and match biotransformations using omics data to generate a reaction list. Then we have to balance the charge and the elements in each reaction. Then based on experimental measurements, we have to construct the biomass equations. Usually this biomass equation stands as the objective reaction or the reaction in which we want to maximize. After this was done, network connectivity analysis is done to ensure that no gaps are present in the network. And finally, all of this are put into one particular model, and these are provided as hosted as genome scale models in servers such as the Big G database. So once this summary of this network reconstruction was provided, then we went on to a little bit of mathematics since the stoichiometric matrix and the mass balance involves the mathematics. This is where we discussed the kind of system that we work with, which is a determined system. By determined system, I mean to say that when we have to find, when we have k constraints and m numbered of measurable fluxes, and if we have to find n fluxes, then the value of d, which is equal to n minus k minus m, is whether it's greater than 0 or lesser than 0 or equal to 0. That was a part that was discussed. Usually, in a determined system, the value of d is usually 0, meaning when you have when you have a certain number of measurable fluxes, in our case m, and certain constraints, in our case k, then we can easily find the value of the remaining. But in our case, which is a metabolic network, it's an underdetermined system because we don't realistically know all of the constraints that are involved. So D is usually greater than 0 because uh, the number of fluxes that we need to find is usually more than the number of measurable fluxes. And at this juncture, I would want to measure that mention that all these methods for flux optimization, they do not describe how the certain flux distribution is realized. Meaning how, as in like, we don't know how much of an enzyme is present in, in a system. So because we don't know how much of an enzyme is present, we don't have an idea of uh, why a particular flux has been is for available for a particular cell. But what we know is that the flux distribution, which is the list of all the flux values for all the reactions that we get from this optimization method, is optimal for the cell, which produces the highest rate of biomass. Then moving on, the different kind of constraints, such as the thermodynamical constraint, which talks about essentially the reversibility of a reaction, then maximum enzyme capacity, the mass balance, and kinetics. These are the four different constraints that are usually applied. Once the constraints were discussed, then the kind of solutions that we can obtain from uh, these optimizations was discussed. So majorly, the kind of solutions are either single solution, where we have one particular fixed set of value, uh, or a degenerate solution, where we could possibly have more than one values. So this was also mentioned. Then moving on, we were introduced to different um, analytical procedures that would help in mathematically understanding the capacity of reactions within a system. So the first one that was discussed is the flux variability analysis, or FVA. So here, what was discussed is, for a given optimal state, one can find the maximum and minimum allowable flux that a particular reaction can have while still supporting the functional state of the network. This is FVA. So what it means is that we will ensure that the objective reaction, which is the biomass in many cases, is maximized. But also we will ensure, or we'll put a constraint to ensure that our reaction of interest is maximized or minimized. So through this method, we will be able to get a unique solution where our reaction of interest is also maximized, the biomass is also maximized. The next point that we want to discuss is the flux coupling. This is what Ignatius had mentioned. I would just like to see if the lecture notes are available for this. If yes, we can go check that. Uh, am I audible, Ignatius? You there?
Yes, sir, yes.
Yeah. So this is in week 4, right? Uh, yeah. So Ignatius, you can start with your doubt. So this is what the slide for flux coupling. That's what we see here. My screen. Oh, my screen. Wait. Let me change it to. Let me present that. Yeah. Are you able to see the slides that were used for the lectures?
Yes,
Types
sir,
of
clearly.
coupling. Yeah. So you can post your question. Is your
Yeah, yeah,
yeah.
sure.
Huh. And then we'll revise this concept and try to address your
Yes,
doubts.
sir.
Yeah.
Yeah, no, so my question was this: how do you derive like which is fully coupled, which is directionally coupled, and which is uncoupled from the reaction map that they've given? Like, like what is the criteria? I mean, by definition, I know like if one depends on the other, then it is directionally coupled and so on. But like from this given reaction map, how do you find out which is fully coupled and directionally coupled?
Okay. So the first thing that we need to know here is the values of the different fluxes, whatever we are dealing with. Once we know the values of the different fluxes, here it's defined as what is Rmin and Rmax, which basically gives you the minimum value of the ratio of the fluxes and the maximum value of the ratio of the fluxes. So since we know the minimum and the maximum values that this can possibly take, those are set as constants. In directionally coupled reactions, usually both, usually this R minus zero. If one of it is zero, the other would also be zero. And R max is a constant. If it's partially coupled, what they say here is that it need not be zero and it could be in a range between a particular value C1 and C2. In a fully coupled reaction, there is no uh, there is no possibility of how do we say one particular reaction being zero because both reactions are dependent on each other. The value of Rmin and Rmax are the same. So there is no maximum. If one increases, the other flux value also increases proportionately. So the ratio of them remains a constant. And whereas in an uncoupled, in a directionally coupled reaction, they say that because both of these are aligned towards producing a specific product, two reactions aligned towards producing a specific product, and because none of them can be zero, the R minimum itself is C, and the maximum can reach up to the theoretical limits of infinity. Whereas in an uncoupled reaction, because they realistically do not interact, the R min is zero. But I am unsure why the R max is infinity in uncoupled reaction. I am not sure about the uncoupled reaction having R max equal to infinity. But otherwise, what you need to understand is you need a minimum information about the fluxes these reactions carry out, from which you get the R min and R max, and that is how you try to say whether reactions are coupled in a particular manner or not. If it's not clear, we can again go through it and I'll try to rephrase my answer. You can tell me.
No, sir, what you said was clear, but so based on the reaction map alone, we won't be able to find out that whether it's coupled or directionally coupled. Is that what you're saying?
Okay, from the reaction map gives you an idea about how they are interconnected. That is needed. Apart from this, you need to know the values of the fluxes as well. So both that information is needed. You can't be sure about the type of coupling just based on the reaction map alone or just based on the flux values alone. Only when you have the reaction map and the flux values, you get an idea of whether they are coupled in a particular manner or if they're uncoupled at all.
Okay, sir.
Like, for example, let's consider two different fluxes here, like V1 and V7. Uh, Whatever flux that goes through as V1, definitely it comes out as V7 or goes back as V1, negative V1. That's the only way V1 and V7 are connected. That's the only way V1 and V7 are connected. So in this case, maybe it is straightforward enough to call it fully coupled, independent of the flux values. But if you consider, for example, let's say uh, V V6 and V3, you know that both of these produce C. These fluxes produce C, but you don't know the origin of the fluxes. So only if you know the reaction map, you will be able to get the origin of the fluxes. And you see that V3 and V6 are fluxes for from B to C and D to C respectively. So they are not linked at this particular juncture, meaning that it is independent of each other, which is why it is given as uncoupled here. Uh, did that answer your question in any manner, or we can discuss that if that doesn't?
No, no, I got it.
Okay, yeah. yeah, once this flux coupling was discussed, then the idea of dynamic flux balance was introduced, which is answering the question of how does the concentration of a particular metabolite varies over time. So while this concept was being introduced, two different kind of reactors or large scale synthesis of uh, large scale microbial synthesis was discussed. One is the batch method, the other is the continuously stirred tank reactor. So in the batch method, we have a particular environment in which a microbe constantly grows. There is no supply of media. One there is only one initial time point in which media is supplied, which is the initial time t is equal to 0. Then slowly the microbe grows into different growth phases, and here the exponential growth has a constant growth rate. So as the microbe grows, it consumes the media and the concentration of the media reduces. This is one particular type of reactors. The other is the continuously stirred tank reactor. This is a special case where there is a constant supply of the media into the reactor system and a constant removal of the cell biomass from the system. So this is ideally modeled to be in steady state, where the concentration of all the uh, components within the system is constant. So this was discussed in dynamic flux balance analysis, just to give an idea about uh, what are the kind of experiments that are needed to validate the mathematical models. So here, um, integration of a particular variable at two different time points was discussed with the growth rate as constant, like we had discussed in exponential growth phase for batch cultures. The other algorithms that were discussed as a part of this lecture, that week's lecture, is the gene deletion algorithms, where we try to simulate knockouts and MoMA and room. So, in a particular knockout simulation, either the flux through that particular reaction is set as a constraint to 0, and then the objective is maintained, and the flux distribution for all other reactions within that model is then calculated. This is how knockouts are simulated. A particular concept of interest for this constraint based approach is MoMA. MoMA is expanded as minimization of metabolic adjustment. So when we delete genes, the cell might not always want to maximize its growth rate. but instead it would uh, minimize its metabolic adjustment. So basically, the cell would adopt a new metabolic state that is compliant with the genetic intervention or the new constraints. This is MoMA. And here, uh, two different states in which the cell exists is assumed. And using quadratic programming, under the deletion conditions, we want to minimize the distance between the flux for the objective and the flux for the objective under the same wild type conditions. So what we need to understand is that when we are deleting genes, the organism may not be able to grow as freely as it was earlier. So what we want to see is that whether the organism finds a new distribution that is minimally different from the old distribution. And this is done using quadratic programming. So far we've been doing linear programming, but here quadratic programming was introduced. Then room. Room is regulatory on-off minimization. So this tries to predict the metabolic steady state following the adaptation to a perturbation by minimizing the set of regulatory changes. So here we set a threshold for how much the flux can change, and if the flux change is beyond the threshold, then the particular reaction flux is considered for minimization. These were the different kinds of algorithms that were discussed. So once the different algorithms were discussed, then the different parameters that can be calculated for this analysis was also discussed. Two are of specific mention. One is the shadow price, the other is the reduced cost. One is shadow price. So in shadow price, what is uh, uh, calculated is if there is a particular uh, reaction that we are interested in, to what extent can the flux of other reactions be altered? And what effect will it have on the cellular processes such as growth and product formation when we alter a particular reaction? This is what is understood from the shadow prices. So this is basically calculated by taking the derivative of the objective function at the boundary. This will also help us determine this will also help us determine whether the cell is limited by a particular constraint. Next, reduced costs. So here, what we get is that, let's say there is an enzyme that was previously not active. Now, if we are forcing that particular enzyme to be active such that the reaction carries a flux, then how will the objective function be reduced? This is calculated in the reduced cost. This is a sensitivity measure. Sorry, there was an issue. I'm just continuing. So, what we were talking is uh, about shadow prices and reduced costs. Then, moving on to something called as extreme pathways. So, extreme pathways are unique and minimal set of basis vectors that can describe all different possible phenotypes in a non-negative linear combination. So let us say we have a stoichiometric matrix. Now, this matrix will have many fluxes that are yet to be found. So, if we find a way in which all the fluxes in this particular stoichiometric matrix can be defined by combination of few number of fluxes, then these few number of fluxes itself would be sufficient to describe the set of uh, reactions for this stoichiometric matrix. So this entire network can also be redrawn based on these two fluxes alone. So the coefficients of these fluxes along with these fluxes is called the basis vectors. But one thing that needs to be taken into consideration while talking about these flux basis vectors is that since we are trying to get a relation between different fluxes in a particular reaction by arbitrarily taking fluxes based on the stoichiometric matrix, these need not be always right. So further validation is required to identify the right set of basis vectors that can define an entire pathway. Apart from this, elementary modes, which are the unique set of non-decomposable enzyme sets that characterizes the convex solution clone, were also introduced. Further moving on to 13C MFA. This is the 13 carbon isotope metabolic flux analysis, where we have a 13 carbon labeled substrate. Then we introduce this to a cell and we analyze the intracellular metabolites with GCMS or NMR. A direct interpretation of this data can help in identifying the active pathways in a given network. In a 13 carbon labeled substrate, because 13 carbon labeled substrate can be decomposed into because the 13 carbon labeled substrate can be decomposed into multiple different precursors, we might get different isomers of the metabolites having different fractions of the labeled carbon. These isomers are called as the isotopomers. Now, the flux distributions are first inferred from the metabolic network connectivity and atom mapping information. And by applying the concept of steady state mass balance on each isotopomer, we get an idea about their distribution. Generally, this is done by involving a recursive algorithm that first tries to predict the amino acid labeling, then it compares with the experimental data and chooses the fluxes. This iteratively occurs until we minimize the sum of the squared deviations between the predicted and measured mass distributions and fluxes. So there are a few challenges associated with this 13C MFA. The first is that um, steady state culture. So there is a trade-off that exists between label carbon in a bioreactor where steady state can be achieved and using shake flasks where little labeled media will be used. But steady state is difficult to be achieved in shake flasks. The other challenge with respect to this 13C MFA is that it cannot analyze the entire metabolic network of a given organism. Rather, only simple metabolic networks can be analyzed using 13C MFA. Also, when there is co-culturing or mixed culturing, where more than one particular organism is cultured together, then because 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 we won't know which metabolite came from which organism, it is challenging to identify or analyze using 13C MFA. Also, there are computational bottlenecks associated with 13C MFA since 13C MFA does not always have a straightforward algorithm to be able to calculate the fluxes.